Dear friends, welcome to Botany A to Z. On this channel, we upload videos on topics from botany and other life science subjects like biotechnology, biochemistry, genetics. So if you are preparing for competitive exams, please subscribe our channel. And if you are more convenient with Hindi or Urdu language, then you can watch another video with the same contents, with same topics, with same pointers that has been uploaded in Hindi Urdu language. As far as today's topic is concerned, today our topic is difference between replication in bacterial cell and replication in eukaryotic cell. We'll be focusing on all aspects like origin, enzymes used, primases, topoisomerases, DNA polymerases, the termination, direction, speed, rate, and primer removal, and other factors. Everything will be dealt in detail. We'll dig in deep so that you get clear concept of replication in bacteria, replication in eukaryotes, and differences between the two. First of all, let me talk about origin of replication. There is only one origin of replication in bacterial prochromosome. The replication starts only at one point. It is bidirectional. The two focus move in opposite direction before they meet at termination sequence and replication is accomplished. Since the genome size in E. coli is small, it is in bacteria is small. For example, in E. coli it is 3.9 into 10 raised by 6 base pairs. Since the rate is high, the speed at which the nucleotides are added, the processivity of the DNA polymerase is high in E. coli. Therefore, only one origin of replication is required. It is in comparison to eukaryotes, wherein you have a large genome size. For example, in humans, the total genome size, the size of the DNA content present on the 46 chromosomes, that accounts for about 10 raised to power 9 base pairs. That means more than 1,000 times greater than Escherichia coli. And the rate of the enzymes for adding nucleotides is about 100 per second. So since the rate is low, since the genome size is large, so multiple origin of replication are required. Every chromosome has many origin of replication. The replication is bidirectional as in prokaryotes, but it starts at many points so that many forks are formed simultaneously and the replication forks move in two directions, and every replication fork meets its adjacent fork. As a result, replication is completed in less time than it would be if there was only one origin of replication in such a large linear chromosome. Despite this, it takes 8 hours for whole genome of human beings, for example, an eukaryote, whole genome of human cell to replicate in comparison to eukaryote to Escherichia coli, a bacteria, wherein whole genome is replicated only in 42 minutes. Although there is only one ORI, but the high rate and less genome size accounts for this small duration at 42 minutes. The ORI, the single ORI in bacteria, is usually about 250 base pairs long. For example, in Escherichia coli, it is about 258 base pairs, and it comprises of 30 nucleotide long repeats called 13-mers, many repeats of these 13-mers, and other repeats called 9-mers. These 13-mers are usually AT rich. It is these repeats wherein the replication machinery enzyme get binded. In eukaryotes, the origin of replications are complex, although the size of every individual ORI is not that large, but they are complex in terms of the sequences. And usually ARS is used for ORI, for origin of replication in eukaryotes. For example, yeast ARS, autonomous replicating sequences. Another difference is that we can see a theta-like structure, a theta-like intermediate during the replication of prochromosome in bacterial cell, but that can't be seen in eukaryotic cell. It is because since there is single ORI, since replication starts only at one point and it is bidirectional, and the chromosome and the prochromosome in bacteria is circular, so when the replication is halfway down, when half of the replication has completed, the structure, the parent DNA and the half newly formed daughter DNA, the structure looks like Greek letter theta. This can't be observed in eukaryotic cell. Since DNA polymerases, as you know, can't synthesize de novo, they require RNA primers and RNA primers are synthesized by enzymes called primases. In bacteria, in eubacteria actually, there is a separate primase called DNase G for synthesizing that small oligonucleotide primer. This enzyme is a monomer. It synthesizes few nucleotide long RNA primer. In comparison to this, in archaebacteria and eukaryotes, there is a different family of primases called Eukaryotic Primase AEP family. It is different from DNase G. For example, in eukaryotic cell, the DNA polymerase alpha, actually so far there have been discovered 5 main DNA polymerases in eukaryotic cell, and of these one is DNA polymerase alpha. The DNA polymerase alpha actually acts as primase. There's no separate primase like in bacteria DNA G. In fact, DNA polymerase alpha itself acts as primase. DNA polymerase alpha, when you see this structure, DNA polymerase alpha is actually a dimer, a heterodimer. It has one subunit which is RNA polymerase in nature, and another subunit which is DNA polymerase. So the term DNA polymerase alpha actually accounts for a dimer, a heterodimer, in which first subunit is RNA polymerase and the second is DNA polymerase. It is this RNA polymerase subunit that synthesizes the small RNA primer, and that is followed by the synthesis of DNA by this DNA polymerase subunit. The main replication enzyme after primer have been synthesized in prokaryotes and eukaryotes, then there is the phase of elongation. So the main replication, the main polymerization enzyme that accounts for the elongation of daughter strand is DNA polymerase III in prokaryotes. In prokaryotes, so far, 3 main DNA polymerases have been discovered, first, second, and third. And it is DNA polymerase III that is the main replication enzyme. It has high processivity, it has high rate. It is the enzyme which carries out the actual replication process, the elongation process. It is a large, complex enzyme comprised of 10 different subunits. And these 10 different subunits actually form 2 major dimers, 2 slightly different dimers. Here you can see. So there are many subunits in this DNA polymerase III hollow enzyme. They form 2 components. This is one core component and this is another core component. They are more or less similar, except for the fact that one of the component has gamma complex here, the clamp loader. We'll discuss it in detail in the next slide. So one of these components has gamma complex. These 2 components of the same hollow enzyme carry out the replication of leading and lagging strands. So it is single enzyme with 2 major core components comprised of 10 different subunits that carry out the replication of leading and lagging strand in prokaryotes, as you can see. So one component, this is another component of the same DNA polymerase III hollow enzyme. In eukaryotes, there are 2 different enzymes, DNA polymerase delta for lagging strand and DNA polymerase epsilon for leading strand. As I said in the last slide that DNA polymerase alpha, after adding few RNA ribonucleotides with the help of its RNA polymerase subunit, it then adds a few deoxynucleotides because it has second subunit that is a DNA polymerase. But this DNA polymerase alpha has a problem that it lacks 3-5' exonuclease activity. Look, DNA replication needs to be error prone. The enzymes should have exonuclease or proofreading activity so that they can look back and correct any possible error. But this DNA polymerase alpha lacks 3-5' exonuclease activity and its processivity is very low. So DNA polymerase alpha is good for initiation, for initiating the replication, but not that good for elongation. It is replaced by DNA polymerase delta on lagging strand and DNA polymerase epsilon on leading strand. This is called polymerase switching. So, attacker of processivity. DNA polymerase third in prokaryotes and DNA polymerase delta and epsilon in eukaryotes are main polymerization enzymes, main replication enzymes because of their high processivity. Processivity means that how long a DNA polymerase can remain attached, can remain loaded on parent strand, and how many nucleotides it will add in a unit time. If a DNA polymerase is unable to keep loaded itself on the parent strand for a long time, it will detach over and again. As a result, the rate of adding nucleotides will be low. So enzymes need to have, DNA polymerases need to have high processivity. That means they should remain loaded on the parent strand for longer durations and add nucleotides quickly in one strand. DNA polymerase third and DNA polymerase delta and epsilon have good processivities. DNA polymerase third, the high processivity in DNA polymerase third in Escherichia coli in bacteria is because of beta clamp. So beta clamp is the processivity factor. You can see this beta clamp. Beta clamp is the processivity factor in eubacteria. And this beta clamp is loaded on the parent strand by gamma complex. So processivity factor in E. coli is beta clamp, and the clamp loader is gamma complex. As you can see here, this gamma complex loads the beta clamp of both the components on parent strands. In comparison to this, archaebacteria and eukaryotes, not only eukaryotes but even archaebacteria, have different processivity factor called pcna, proliferating cell nuclear antigen. And this processivity factor pcna is loaded on parent strands by replication factor C. So here the clamp loader is replication factor C. As you know that when the helicase enzyme and other replication machinery moves forward in the form of replication fork, the twist, the stress is passed on to ahead of the replication fork in the double-stranded parent DNA strand. Since the replication fork is moving, since the helicase is unwinding the double-stranded DNA, as a result of the stress, the supercoiling moves ahead of this fork and results in over twisting or over supercoiling of parent strand ahead of replication fork. And this over supercoiling is not good for not good for smooth replication, because you need to have relaxed double-stranded DNA so that enzymes can have easy access to it. In order to remove this extra supercoiling, this stress ahead of the replication fork, there are enzymes called topoisomerase first and topoisomerase second in eukaryotes. The difference between topoisomerase first and second is that the topoisomerase first makes a cut only in one strand in order to remove the stress, in order to remove those twists from the double-stranded DNA. Topoisomerase second makes cuts in both strands in order to remove the stress. In prokaryotes, there is a slightly different enzyme called gyrase, and it carries its function in the same manner as DNA polymerase second. Primers, removal of primers. Since there are primers formed during the replication and they are RNA in nature and they need to be removed, DNA polymerase first in prokaryotes is unique in that it has 5-3' exonuclease activity. As you know, most of DNA polymerases have 5-3' polymerization activity. 5-3' polymerization activity, that is, they add nucleotides in 5-3' direction. Plus, most of them have 3-5' exonuclease activity. That is, they remove incorrect nucleotides in the opposite direction, in 3-5'. They look back and remove those nucleotides. For that, they require 3-5' exonuclease activity. So most of the enzymes have 5-3' polymerization activity and 3-5' exonuclease activity, except DNA polymerase first. It has not only 5-3' polymerase activity, 3-5' exonuclease activity, but it has also 5-3' exonuclease activity, that is in the same direction as polymerase activity. This 5'3' exonuclease activity is required, is used by this enzyme to remove primers and fill the gaps. Why it is 5'3' polymerase activity. In eukaryotes, there are special enzymes for removing primers. It is RNase H and flap endonuclease, FEN1. RNase H, as you know, the primer is simply an oligonucleotide, a ribonucleotide made of few ribonucleotides. So RNase H removes most of those ribonucleotides, and the last one is removed by FEN1. It is flap endonuclease. That is in eukaryotes. Since the genetic material, the chromosomes in eukaryotes are linear, and as you know that DNA is double-stranded and antiparallel, there are 5'3' strands that are opposite to each other, 5'3'1 and another is 3'5'. And the primers, as we saw, that they are removed, but opposite to the 3' end of parent strand at one end, opposite to the 3' end of parent strand, there remains a RNA primer. When it is removed, there remains a gap actually. Because when this primer will be removed, there will be nothing to extend. There will be no DNA nucleotides available that a DNA polymerase can work on and extend it to fill the gap. So when the last primers, the primers are opposite to the 3' end of parent strand, 3' end at one side and 3' end at opposite end. So when the primers opposite to the 3' ends of parent strands are removed, there remains gap in this linear chromosomes. And that gap can't be filled by simple DNA polymerases. That gap is filled by special enzymes called telomerases. So it is telomerases that carry out the replication of telomeres, the ends of DNA molecules. These telomerases are actually ribonucleoproteins. They have in them an inbuilt RNA strand. That RNA strand is used by the protein component. It has a RNA component and a protein component. The protein component, the enzyme component, uses this RNA strand as template and fills the gap against the 3' end of parent strand, completes that daughter strand. So because it uses RNA as template, its own RNA as template, so in nature it is reverse transcriptase. Using RNA as template, it synthesizes DNA. Since there are no telomeres in bacterial chromosomes, bacterial prochromosomes, they are circular, so no telomerases are required. In fact, the replication for the bidirectional, since the replication is bidirectional, the two focus meet at special sequences called TER sequences, and with the help of TUS proteins, the replication machinery, the enzymes are taken off, and replication is completed in this way in bacterial cell. So I hope you have got very much acquainted with the differences between bacterial and eukaryotic replication. If you like the video, please share the video, and if you have not subscribed to the channel, please subscribe Botany A to Z. Thank you.